So in this video, we are going to be looking at a cell cycle and mitosis. Just to refresh your memory on the cell theory states that every living organism consists of one or more cells. A cell is the smallest unit of life, individually alive, even as part of a multicellular organism. All living cells come from the division of pre-existing cell, and that's the part that we're going to be focusing on in this video. We're going to see how exactly does a cell divide and create two genetically identical copy of itself. And finally, cells contain hereditary material, which we know now it's DNA, which they pass on to their offspring during division, and they're able to do that because they would copy all their DNA, right? So in the previous video, we learned about DNA replication and the mechanism of how DNAs are being copied. So once you've copied all the genetic material, now you can pass it on to the cells that you are going to be dividing into. But first, what is the purpose of cell division? We use cell division to replace lost or damaged cells. If you cut your finger, some cells would have been gone or damaged, and as the wound heals, we're going to have to need new cells to replace the lost or damaged ones. We also use cell division for growth and development. Think about when you were just a baby, right? There were fewer cells in you compared to the adult version of you. So all that extra cell came from cell division. Some organisms use cell division as a way for reproduction. Mitosis can be used as a form of asexual reproduction. Mitosis is basically cell division, okay? Cell division. So what is asexual reproduction? Asexual reproduction is a type of reproduction that does not involve fertilization of an egg by a sperm. Okay, so the advantage of that is you only require one individual to do so. Right? If you think about things like an amoeba, which is single pawn organisms, it would be difficult for them to find perhaps another amoeba to mate with, right? So they do not need a partner. They can just divide themselves using mitosis, and they are going to be able to clone themselves and create many copies. So it's quite fast and require less energy. A lot of organisms can do that, not just unicellular organisms. Sea star. Did you know if you cut off an arm of a sea stars, not only can it regenerate the lost arm, but in some cases, if the chopped off part is big enough, it's able to grow into another sea star. So that's an example of asexual reproduction. There's something called a hydra, which is kind of this half plant, half animal microscopic organism, and they grow by something called budding. Okay, so this here is the mother hydra, and then there is a baby one growing off the body. Okay, and when it's big enough, it's just gonna break off. So no partner required, and just one individual is needed, and this is going to be a clone of itself. So that's asexual reproduction. Same thing for your plants, right? If you have any plants at home, things like African violet or even things like aloe vera, you can break off a branch and then the plants can just kind of clone itself, right? So the major disadvantage of this asexual reproduction is there's low genetic diversity because you are essentially copying all of the genetic materials, right? So every individuals from that original cell are going to be identical genetically speaking, and so if there is some kind of undesirable trait within that genetic material, then it's going to show up in all of the copies from that point forward. By now, we've spent quite a lot of time talking about DNA, and I'd just like to remind you that DNA is the genetic material that makes up the chromosome, but DNA doesn't always exist in the form of chromosome. Okay, if a cell is not dividing, then the DNA actually exists in the form of chromatin. Okay, these are DNA that has been wrapped around histone proteins that enables them to kind of coiled up into these chromatin, which will be able to fit into the nucleus of the cell. When the cell is ready to divide, after you copy all the DNA genetic material and it's ready to divide, they would condense even further. And when you take the chromatin and you coil them up even further, then you end up having chromosome, okay, which is the coiled and condensed version of chromatin. So in humans, we have 23 pairs of chromosomes, a total of 46. Of the 23 pairs, 22 of them are considered as autosomal chromosomes, or simply autosomes. These are chromosomes that are going to be determining various traits we have. So things like your blood type, your skin color, your type of hair you have, curly hair, straight hair, right? And you know, whether your earlobes are attached or detached. So many details are going to be encoded in these 22 pairs of autosomes, except for sex. Okay, the last pair, the 23rd pair, is considered as the sex chromosome. So that's the one pair that will be determining the gender of the individual. So for male, we will be having one X chromosome and one Y chromosome, and for females, females will have two X chromosomes. So of the 46 chromosomes, 23 will be inherited from mom, and the other 23 will be inherited from the father, right? And so they come together and form 46 chromosomes in total. The number of chromosomes seems to be not related to the size of the organism. We have something like a giraffe would have 30 chromosomes, even though it's much bigger than us humans. They have less chromosomes. And then on the other hand, you have something like a dog. It has 78 chromosomes. Okay, and the size of a dog is smaller than human. So there isn't a correlation between how big the organism is and how many chromosomes they have in their cells. So all cells goes through what we call the cell cycle. Right? The cell cycle involves two major phases. The first one is interphase. In interphase, the cell will be performing its normal functions. So if a cell is supposed to be part of a gland, then it's going to be making proteins and secrete them. If cell is a muscle cell, then it's going to be responsible for producing contractions and so on and so forth. And during the interphase, in addition to performing its normal function, these cells will also grow and prepare for mitosis, which is the actual cell division. Right? So in mitosis, one parent cell is going to divide and become two genetically identical daughter cells. Here in this diagram, it shows the entire cell cycle, and you can see the interphase, which starts here all the way to here. That takes up roughly 90% of the entire cell cycle time, if you would, and the cell will only spend about 10% of its time undergoing the mitosis. So this is just a rough estimation, I guess, because it depends on the cell type you're talking about. Some cells would spend much longer on the interphase than others. So things like neurons, brain cells, right? They actually don't divide at all, so they will be spending their entire lifespan in the interphase without going into the mitosis. Whereas something like skin cells, they actually divide quite rapidly, and so they will cycle through the cell cycle much faster. But typically speaking, a cell will spend 90% of its time in interphase and 10% of its time in mitosis. And you can see within the interphase there are different stages as well. In G1, the cell will be growing. In the S phase, that's where the DNA replication will take place. So everything we learned in the previous video, with the helicase and gyrase and all this stuff, that happens in the S phase. And then you would have a G2 phase where there's going to be more growth and final preparation for division. And then in mitosis, there are also different phases in the actual division process. So In mitosis, in interphase rather, this is what the cell looks like in interphase. There is the blue here shows the nucleus, and the green just shows some of the cytoskeleton, and the red is kind of like a background stain that shows you where the cell is. And in interphase, this is where the cell spends most of its life, and it performs regular function, as we said. But near the end of this phase, the cell would undergo mitosis, and the DNA would duplicate and replicate itself. So you can see here in this cartoon picture, the chromosome, the DNA rather, in the nucleus, it's now in the form of chromatin, which is DNA around histone proteins, and the nuclear envelope is still intact. And you might remember these centrioles that we learned in lecture 3. They are the ones that are going to be creating the spindle fibers, the microtubules, that will be guiding the movement of chromosomes once cell division starts to occur. Here are 2 chromosomes. Remember, we have 23 pairs of chromosomes. Half of it is going to be inherited from each parent. So we have the green one here, maybe inherited from dad, and then here we have another one that's inherited from the mother. And so right before mitosis, we are going to have to do DNA replication. So after replication, you will have 2 copies of the father one, and then you'll have 2 copies of the mother maternal chromosome. Okay, and they're going to be held together at this special location, which is called the centromere. Okay, and the identical copies that you've created, they are called sister chromatids. Okay, sister chromatids. So just to quickly say it one more time, this is the paternal chromosome that you inherited from your father. This is the maternal chromosome that you've inherited from your mother, and this would be in your cell. Okay, so if this cell wants to divide, it would have to clone each of these chromosomes. So it will do the DNA replication process, and now you will have 2 of each copies, and the identical copies are referred to as sister chromatids. Now that you have copied all your DNA genetic materials, the cell will be ready to perform mitosis, the actual cell division. And there are 4 phases involved in mitosis. There is prophase, metaphase, anaphase, and telophase. In early prophase, the chromatin will coil up into visible chromosomes. So you can see the individual chromosome strands here. If you take a closer look, they kind of look like a bunch of coiled up spaghetti. If you go back to the earlier slide here in interphase, you cannot see the individual chromatin. That's because chromatins, even though they're kind of coiled up, they are not as condensed as the chromosome. So they won't be visible under the microscope, whereas the chromosome, you will be able to see them here. By now, the DNA has been replicated, and DNA that has been replicated are going to be held together at the centromere, and these 2 copies, identical copies, are going to be called the sister chromatids. Okay, so chromatids with a D are the exact copy of the DNA, whereas chromatin, that's the uncoiled version of the chromosomes, which is the natural state of the DNA when a cell is not dividing. Right, so don't confuse the 2. At this point, the nuclear envelope is going to start to disappear. Okay? It's going to dissolve so that the genetic material can come out, the chromosome can come out into the cytoplasm, and you can move them around and guide them in the next part. And the centrioles are going to start to migrate to the opposite end of the cell, and as they move towards the opposite end of the cell, they will produce these spindle fibers. At later part of the prophase, the nuclear envelope is going to be completely dissolved, so the chromosomes are going to be out of the nucleus by now. You can see that the green here is showing the location of the centrioles, and the spindle fibers are going to be attached to the centromere of each chromosome pair. Okay, so there is a centromere, and you can see the spindle fibers is attached to it. Okay. Next, we have the metaphase, right? M for metaphase and M for middle. Right? In metaphase, the spindle fibers are going to kind of engage in a tug of war on the chromosomes, so that eventually they will all lined up in the middle. Okay? So here it says the spindle fibers lines up each pair of sister chromatids at the center of the cell. Remember, this copy here is the exact same copy as that one. Okay, and then this copy is the exact same copy as that. So this is a sister chromatid, and this is a sister chromatid in this pair, and then this is a sister chromatid, and this is a sister chromatid. And they are going to be held together at the centromere. And what's important here in metaphase is that the spindle fibers will pull back and forth on these sister chromatids until they all lined up in the center of the cell. And the next phase is the anaphase. A for anaphase, A for apart. In anaphase, you're going to be pulling the sister chromatids apart. Remember, this copy is exactly the same as that one, so you want them to migrate to the opposite pole in the cell. So when you ultimately divide the cell, each half of the cell will have the exact same genetic material. So the spindle fibers will pull them apart, and it will pull them towards the opposite pole of the cell. And the last part is telophase and something called cytokinesis. Telophase is basically going to be the exact opposite of what happened at prophase. The chromosomes will now start to uncoil back into chromatins, and the nuclear envelope will start to form again to surround the genetic material, and now that the spindle fibers has finished its job guiding the chromosomes to the opposite end, we don't need them anymore, so we're going to be breaking them down. And towards the end of the telophase, we're going to have the splitting of the cytoplasm, and that is what cytokinesis means. Okay, so there is going to be something called the cleavage furrow that's going to form in the middle of the cell, and it's going to cleave the cell in half, and that's how you end up having two separate cells, each with their own genetic material that are going to be identical to the original cell. So here are some onion cells in the root tip, and perhaps we can try to identify what phase each of them are going to be in. So maybe pause the video and try it yourself, and then you can continue to watch to see if you got it right. So this one, right, you can see this cell, all the chromosomes are lined up in the middle, so we know this one must be metaphase. Okay. And over here, we can see the chromosomes are being pulled to the opposite end, so this one is in anaphase. Okay, so you can see another one in anaphase here, right, and then another one in anaphase here. This one, it looks like the chromosomes are very concentrated at the poles, and if you look really closely, you can start to see that there is a new line forming in the middle, which tells you that the cells is about to split in two. So this must be telophase. And over here, we can see that we can see the chromosomes, but they're not lined up or being pulled to one end or the other. So this must be prophase. And if we look at something like this, this one we cannot really see the individual well-defined chromosomes. Just kind of like a sea of blue in the middle. So this one must be in interphase. Okay, where it's not dividing. Okay, remember when it's not dividing, you cannot see the chromosome. They are still in the chromatin form. So that's all the phases in mitosis. Now, the cell cycle actually has built-in checkpoints that allows problems to be corrected before the cell cycle advances. You might remember earlier in the interphase, there are three separate parts. There is the G1, and then there's the S phase, and then there's G2 before it goes into mitosis. So G1 kind of checks for the quality of the DNA to make sure that it is good enough for DNA replication to take place in the S phase. So we talk about how sometimes the telomeres get critically short, and then the cells will not be allowed to divide. So that's something that will be checked in G1. Okay, if a cell sees that its telomeres are too short, then it will not be allowed to progress through the cell cycle anymore. And then once you've copied all your DNA, G2 will kind of check for the quality of the DNA that you've copied, right? Did you make any mistakes? And if there are mistakes, then are you going to be able to fix these mistakes, right? If you are able to fix it, great, you can progress to mitosis, cell division. But if the problems remain uncorrected, then these checkpoint proteins will tell the cell to undergo self-destruction, right? And a programmed cell death or cell destruction is what we call apoptosis. On rare occasions, the control over the cell divisions are lost. Okay, those checkpoints, those G1, G2s are actually mostly protein products that are doing the checking. So you know by now that the proteins themselves are encoded within the genetic material, right? So if there is some kind of errors in the genetic code, that could cause those checkpoint proteins to not function properly. And if those checkpoint proteins are not working, then they might let a cell to progress through the cell cycle despite the fact that it has some critical errors in it. And so all the descendants from that cell are going to contain the same error. And if more errors accumulate, what's going to happen is the cell can now proliferate in an uncontrolled manner, and that creates what's called a neoplasm. Okay, so a neoplasm, it's an accumulation of cells that lost control over how they grow and divide. So neoplasm will result in a tumor, and not all tumors are going to be cancerous. Some tumors are just benign, and you can remove them, and for the most part you're going to be okay. It's when the tumor is malignant, then it's going to be cancerous, and that is going to be harming the body tissue because it's going to be taking up all the resources from the adjacent cells. And as you know, most forms of cancers there are no well-defined treatments for it, and it's kind of like the last frontiers of medicine, right, that we haven't been able to conquer yet. So that's it for this lecture. Make sure you're able to describe the process of DNA replication. So it's the semi-conservative model with all the enzymes involved, helicase, gyrase, DNA polymerase 3 and 1, ligase, so on and so forth. Be able to describe the stages in cell cycle and mitosis. So quickly, G1, S phase, G2, and then for mitosis we have the prophase, metaphase, anaphase, and telophase. And also we just talk about how if you have mitotic errors, it's potentially can result in cancerous cells. Thank you for watching.